is called Saccharum barberi. The other one is called Saccharum officinarum. Hope you met. One is wheat, the other one is rice, next one is tomato, the other one is lady's finger. Dr. M.S. Swaminathan produced amber-seeded Sharbati sonara. It is the purposeful manipulation of plant species in order to create plant types. He was one among the 20 people selected by the Times magazine in 1999. Hello everyone, welcome back to session 4 of this very important chapter called Strategies for Enhancement in Food Production. What are we going to study in today's session? In today's session, we are going to study about the mutation breeding. What is plant breeding or mutation breeding? It is the purposeful manipulation of plant species in order to create plant types which are better suited for the environment. And not only that, they give better yield and they are disease resistant. That is the main aim of plant breeding. What are the traits for which the plant breeding is done? Point number one, to increase the yield. What is the purpose of plant breeding? Why is it done? That is very, very important. To increase the yield and also to improve the quality. Improve the quality of the crop. That is the main aim. And third, to increase the tolerance to the environmental stresses. Environmental stresses like salinity, extreme temperatures, etc. And also to increase tolerance to insect pests. All these are the traits for which the plant breeding is done. So what is plant breeding? It is a purposeful manipulation of the plant species in order to create plant types which are better suited for the environment, and not only that, they give better yield, more yield, and they are disease resistant. Right? So as I said, what are the traits for which the plant breeding is done? To increase the yield, very, very important. For a farmer, the yield is very, very important. So the economy of his crop depends upon his yield. So to increase the yield, second one is what? To improve the quality of the crop. So quality also is very, very important. And third, increase the tolerance to the environmental stresses. May it be like salinity or extreme temperature. Then also to increase tolerance to the insect pests. And also resistance to disease causing pathogens. Fifth one, resistance to disease causing pathogens like fungi, bacteria, etc., etc. So all these are the traits for which the plant breeding is done. As I said, it is also called as what? Mutation breeding. What is mutation breeding? It refers to the use of induced mutations for crop improvement. Why is it done? To improve the crop. Very important is to improve the crop, and mutations that are artificially induced by employing mutagens are called as induced mutations. What do you mean by? So mutation breeding, as I said, it is referred to as induced mutation for crop improvement. And how do we define this mutation breeding? As mutations that are artificially induced by employing mutagens are called as induced mutations. Some common mutagens used in mutation breeding are radiation such as UV rays, X-rays and cosmic rays, etc. Chemicals like nitrous oxide, mustard gas, ethyl methane and sulfonate, etc. All these are used as the mutagens to bring about the process of mutation to improve the crop, right? So what is mutation breeding? It refers to the induced mutation for crop improvement. Mutations that are artificially induced by employing mutagens. What are the mutagens that are employed? Usually it might be radiation such as UV rays, X-rays, cosmic rays, chemicals like nitrous oxide, mustard gas, ethyl methane, sulfonate, etc. So here you can see the change in the gene sequence. What is manipulation? You are altering the gene sequence for a better cause. That is what we call it as genetic engineering or gene manipulation. That is what is being done in plant breeding. Moving on to the very other important concepts of this very important chapter. Hope you can see this person. So a very famous person who is man behind the Green Revolution. He is called father of Green Revolution. Green Revolution. Hope you can see this person. He is none other than Dr. M.S. Swaminathan. MS stands for Mankombu Samba Sivan Swaminathan. And he is called as father of economic biology also, and he is also called as father of Green Revolution. He is one of the revolutionaries in implementing the crop breeding techniques, particularly to produce a crop which gives more yield and which is disease resistant, particularly in crops, economical crops like wheat, rice and maize. And he is the man behind this Green Revolution. And one very important thing you should know about this very important person, that is Dr. M.S. Swaminathan. He was one among the 20 people selected by the Times magazine in 1999 for the 20 top influential Asian people, along with Mahatma Gandhi ji and Rabindranath Tagore. He is one among the three. Only three were selected from India, and among those three, one was Dr. M.S. Swaminathan for being selected as one among the 20 top influential Asian people of the 21st century. So that is the credit that goes to Dr. M.S. Swaminathan. Right. So what is the very important technique that is applied in mutation breeding? In mutation breeding, usually seeds and buds, as we call them as the vegetative propagating plants, are treated with suitable mutagens. So here, particularly the buds in the vegetative propagating plants are treated with suitable mutagens, and the plants are raised in the field and self-pollinated. So what is the technique that is applied in mutation breeding or plant breeding? In mutation breeding, usually seeds or buds in vegetative propagating plants are treated with suitable mutagens. The plants are raised in the field, then they are grown in the field and they are self-pollinated. The progeny of the plants are carefully observed to identify and select mutation of interest. So this is how selecting the type of crop which is going to give more yield and also which is disease resistant is very, very important technique in mutation breeding or plant breeding. As I said, the traits for the plant breeding is what? To increase the yield, to increase the quality of the crop, and also to increase the tolerance to the environmental conditions, particularly the extreme climatic conditions, may it be salinity or may it be high temperature, etc. Dr. M.S. Swaminathan produced amber-seeded Sharbati Sonara, that is a wheat. As I said, particularly he was very much involved in producing particularly a crop like wheat. So he was the one who produced amber-seeded Sharbati Sonara wheat by inducing mutations in Mexican wheat variety called Sonara 64 using UV radiations and gamma rays. As I said, he is the man behind this Green Revolution, and he is rightly called as what? Father of Green Revolution in India. As I said, he is one of the revolutionaries in the plant breeding techniques to bring about the crops which give more yield and which are disease resistant, particularly in crops like wheat, rice and maize. Now, examples for mutation breeding are, so what are the examples we are going to take to learn about the mutation breeding? One is called the wheat, that is Sharbati Sonara NP836, rice called Jagannath, then tomato Pusalal Miruti, then lady's finger called Parbani Kranti, resistance to yellow mosaic virus. As I said, the very importance of this plant breeding technique is what? To see that the yield is more and also they're disease resistant, and particularly they are also resistant to what? Insect pests. That is also very, very important. So what are the examples of mutation breeding? The examples for mutation breeding are wheat, rice, tomato and lady's finger. Moving on to the green revolution. As I said, what is green revolution? Production of more wheat, which is where the yield also is more and it is also disease resistant. So what is green revolution? Let us understand what is it. It is a movement launched in 1960s that has increased the food production not only to meet the national requirement but also for export. Very, very important. This green revolution has not only provided wheat production for our country but also we are started exporting even the wheat. Then it was dependent to a large extent on plant breeding techniques to raise high yield and disease resistant varieties in wheat, rice, maize, etc. So what is green revolution? It is a movement launched in 1960s that has increased the food production not only to meet the national requirement but also for the export. And as I said, it was dependent on large extent on plant breeding techniques to raise high yield and disease resistant varieties. As I said, particularly the plant breeding techniques were applied for what? For wheat, rice, maize, etc. So this is about green revolution. This is a very important concept from this chapter which is usually asked in the examination. So here you can see in the beautiful picture like how we are able to produce more wheat production because of this, and I think the man behind this green revolution is none other than Dr. M.S. Swaminathan. So the present day we are able to get so much of wheat means it is due to the plant breeding techniques of this great man, Mr. Dr. M.S. Swaminathan, who is rightly called as father of green revolution. He is also rightly called as father of economic ecology. Father of economic ecology. Father of economic ecology. So this is about green revolution. So wheat, as I said, wheat is one of the crop that was developed through plant breeding techniques, and that is the reason why we are able to produce more amount of wheat, right? And that is how it has led to what we call it as green revolution. So one scientist by name Norman E. Borlaug developed semi-dwarf varieties, and as I said, the very concept of producing high yield and disease resistant varieties in wheat is mainly because of the semi-dwarf varieties of wheat that the International Center for Wheat and Maize Improvement in Mexico, and which was developed there. And varieties of wheat, like I think we come across a varieties of wheat, right? You can call it as a staple food also. Wheat is called as a staple food, and varieties of wheat like Sonalika, Kalyan Sona, and these were selected from the semi-dwarf varieties and which was later introduced in India. As I said, the very concept of plant breeding technique is to produce better variety wheat so that the yield is more and it is also disease resistant. So Norman, as I said, Norman E. Borlaug developed semi-dwarf varieties of wheat at the International Center for Wheat and Maize Improvement in Mexico. And how was it developed? And as I said, varieties of wheat were developed like Sonalika, Kalyan Sona, selected from these semi-dwarf varieties, which were having high yield and also disease resistant. Moving across, the next very important, that is rice. Semi-dwarf varieties were derived from IR8 and Taijung native. So rice, semi-dwarf varieties were derived from IR8 and Taijung native. As I said, semi-dwarf rice varieties were derived from IR8, developed, what do you mean by IR8? Developed at International Rice Research Institute, IRRI, Philippines, and Taijung native one, that is from Taiwan. So the derivatives were introduced in India. As I said, they were introduced in India in 1966. better yielding semi-dwarf, and later better yielding semi-dwarf like Jaya and Ratna were developed in India. So now the rice production has increased from 35 million, which was 35 million tons in 1960, to 89 million tons in 2000. Just imagine the amount of that the rice is produced. So imagine the drastic increase in rice production. It is all because of the plant breeding techniques. So one is wheat, as we already come across how the green revolution was created in producing wheat. In the same way, rice. So the semi-dwarf varieties were developed from IR. As I said, varieties were developed from IR 8, which is developed at International Rice Research Institute IRRI, Philippines, and Taichung Native 1 type from Taiwan. And as I said, later rice varieties like what? Jaya and Ratna were developed in India. So rice production has increased from what? From 35 million tons in 1960 to 89 million tons in 2000. So imagine the drastic increase in the production of rice. So all this is nothing but it is a contribution given due to the plant breeding techniques. Moving on to the next very important crop, which is having a commercial value, that is sugarcane. So here you can see in this beautiful picture sugarcane. You know, the table sugar what we have in our daily life and the sweets what we have in our daily life, everything is made out of what? It is from the sugar that we get from this sugarcane. So there are two varieties of sugarcane. One is called Saccharum barberry. The other one is called Saccharum officinarum. Hope you might, Saccharum means what? Sugar content. So one variety called Saccharum barberry, which was originally grown in North India. And I think all of us know the nodes, more the internodes, thicker the internodes, more will be the content, sugar content in sugarcane. Here one variety called Saccharum barberry, originally it was grown in North India, but had poor sugar content. It had very poor sugar content. But there was one more variety called Saccharum officinarum, which was grown in South India, which had thicker stems and high sugar content. So a cross was made between these two varieties, that is Saccharum barberry and Saccharum officinarum. So a cross between these two varieties and a hybrid variety combining. So usually the combination of these will obtain what? A hybrid, desirable qualities. Like what? Like thicker stem, high sugar content and higher yield. So this is how, and that has been also introduced in North India. So as I said, it was not possible to grow in North India, that is Saccharum officinarum, which had more thick stem and more sugar content. So when a cross was made between Saccharum barberry and Saccharum officinarum, so a hybrid variety was produced which had the desirable quality like the thicker stem, high sugar content and higher yield, and that was grown where? That was grown in North India. Originally it was grown in South India, but even later on it was also introduced in North India. So just imagine with the plant breeding techniques, we are able to produce a good variety of sugarcane which will have more thick stem and which will have more sugar content. So that is what is the revolution that has been done in the field of plant breeding techniques. So sugarcane, as I said. So what about Dr. M.S. Swaminathan, who is one of the man behind the revolutionaries in plant breeding techniques. Particularly, he was very much responsible for production of more wheat and also plant crop like rice and even sugarcane. So sugarcane, as I said, one variety, Saccharum barberry, originally grown in North India, had poor sugar content, and the other one called Saccharum officinarum, grown in South India, had thicker stems and high sugar content. So a cross has been made between these two varieties and a hybrid variety is obtained by combining these two varieties, that is Saccharum barberry and Saccharum officinarum, and a desirable quality was obtained. Like what? What was the desirable quality that is obtained between the cross between these two plant varieties? One is the thicker stem and high sugar content, and which was also able to grow in North India. Hope you have understood all the concepts what I have explained in today's session. So this is a very, very important concept which is asked usually in the examination, particularly about Dr. M.S. Swaminathan and also about the varieties of crops that he was able to develop with his plant breeding techniques, namely the wheat, the rice and the sugarcane. So what are we going to study in the coming session? we are going to study about plant breeding for disease resistance and also what is biofortification. So I'll be back with some more concepts in this chapter. Till then, goodbye and thank you.